So I think scientific research needs to be accessible to non-scientists for a number of different reasons. So one of them, I'm sort of turning this question on its head, that I think people are interested in what's going on around them, and they are interested in things. So people are just basically interested in scientific research. I also think in the kind of research that I do, is medical-based research, and people care about their health, the health of the people that they know and love, and so people are eager to want to understand about these types of issues. So I think people really want to know about scientific and medical research for those very reasons. So the typical question I get asked, or the point of view that came from my kind of research, is asking, can you use a very simple model organism? And by that I mean a classic type of animal that you study in the lab. I study the basic fruit fly. The fly is something that you see, if you leave fruit out, you'll see a fly come around. And believe it or not, we can learn a lot about the genes that regulate our behavior and that regulate our brain from studying the fly. And then moreover, in our work, we've been able to actually give the fly a human disease, and the fly can tell us stunning amounts about that. Some of the points that my type of research addresses are, first, that you can use a stunningly simple system to learn really important insight about very complex disease problems. So that you can use organisms as simple as a fly, as simple as a yeast, and come up with the pathways that are involved, that the same pathways that go wrong in human biology or go wrong when you express those genes in those simple organisms like the fly. And I think the other thing that's been amazing to me about the field that I'm in, that I've learned, is that I think we learn a lot more about human biology from studying disease than we do by the classical approach. So by the classical approach, when I started out, the way to do biology was not to create a disease model in the fly, but to, pick a process you wanted to study and knock the genes out and find out how they act. But we've learned so much more by taking the genes that cause the human disease and putting them in the fly. And I just don't know that we would have learned that much that fast if we tried that other approach. And to me, that's been a real revelation in how you can go about just making discoveries. I think the most important thing to know is that it's difficult to predict where the most important or impactful findings will come from, that change things that we think about in daily life as well as medical discoveries. So it's very difficult to know for sure that you're not going to make a discovery from bacteria or yeast, that it has to be, for example, the study of the human brain to tell you all the most important information about the human brain. Okay, so when I think about those things that really were surprising to me in my work, I have to say that two things I think are two moments really stand out to me as stunning. So the first one was when we first put a human disease gene in the fly and then we expressed it. And that fly had an effect that was just so stunningly like what you might imagine if you could give a fly that human disease, what it should look like. And I'm not really sure, to be honest with you, what I expected, but I found that it was just amazing. And moreover, it had many features that you don't normally see in fly mutants. So the phenotype, like many human degenerative phenotypes, was late onset and it was progressive, that is, kept going on. And these same features were there in that fly. That was really surprising to me. I became hooked right on that. I was like, this is the most amazing genetic phenomenon I've seen. It's so stunning. And then I would say the second one comes with a finding that followed up on that was really surprising to me. So, we created the fly model not to give the fly a disease, but to see, could we provide insight into this disease so that it would provide help in understanding how to combat and interfere with this disease. And so we tried this one crazy idea of putting this other gene in that we thought, well, maybe this will have an effect, but I don't know. It was just a really long shot. And when we put that gene in, it completely cured the fly. And I was just stunned by that, to think that the animal could tell us that. And that really led me to, the power of the in vivo organism to sort of tell you, what can be the good answers and how you can use this model. I think those were two really exciting points in my life.